regions, it is been given like in different regions or different civilizations, different systems of medicines are being followed. So medicine as a such is a word which is related to about the diagnosis of the disease that is based on the symptoms, and what is the prescription to be given, and how that prescription need to be used. That means how to use the medicine. So the whole thing is called as a medicine. It is a science related to the diagnosis of disease or the problem, and then the like the prescription and the use of the medicine. The whole thing is medicine. So as we know, there is an allopathy medicine, and that is, as it is a process been involved a lot of steps wise, so it is called a system, medicinal system. So the allopathy is a modern medicinal system, and there are different types of medicinal systems are being practiced in different civilizations. So that is, they have been since ancient times. They have been practiced. They are called as traditional systems of medicine. So here in the first unit, we are going to discuss origin and development of different systems of medicine. We all of us know like there Ayurveda, Siddha, Unani, and homeopathy. These are different alternative medicinal systems we are aware of. So we are going to learn about what is the origin of that medicinal system, and what are the plants are being used in that medicinal system, and how important it is for the treatment of different diseases. So here those are the things we are going to understand about the origin of different traditional medicinal systems. Here I'd like to point out one of the thing like here we are going to discuss about Ayurveda, Siddha, Unani, and naturopathy and all. And there is a categorization of these traditional systems of medicine as indigenous systems of medicine and Indian system of medicine. Indigenous means it is applicable to all the systems of medicine being originated in different regions. But Indian system of medicine is the one which has been originated in India, like Ayurveda and Siddha and naturopathy. Okay, so you need to be precise that we had to mention about only Ayurveda and Siddha. If it is traditional systems of medicine, all the systems of medicine which are being practiced since ancient times will come under that category. Okay, so that we'll discuss in detail once we start the topics of that unit. Then the another second unit is of general account of phytochemistry of medicinal plants. Okay, in the beginning, we try to understand how the plants are being used in different systems of medicine. Which are the plants are being popularly been used, we will discuss. Then followed by different medicinal plants will have different components. That is, makes the plant being used for the treatment of a particular problem or a disease. So that is the chemicals which are being found in the plants are generally being referred as phytochemicals, right? So the phytochemicals are nothing but the chemical substances found in the plants. But we have some specification here again. Not all the substances. The substances which are useful for the medicinal property of that plant, they are being more of our concern. So they are often been called as active principles. So here, what is that phytochemistry? What are the components we generally observe in the plants of commonly found in all the plants, which are very specific to certain plants, and what is their significance according to the chemical classification we are going to discuss. Okay, that is about the phytochemistry of the medicinal plants we are going to discuss, which is a very important aspect of the medicinal plants, where we are going to know about what is the chemical nature of that substance. Okay, then about the unit 3 is related to about the medicinal plants. So here we are going to learn about some brief information about the medicinal plants. In the unit 3 and unit 4, we are going to discuss about certain medicinal plants, because normally while we study the botany as a subject, we study different families of angiosperms which are unique in their characteristic features, and some might be even having some medicinal values. But here we are going to study about specifically about the medicinally useful plants. So that means a little more detailed study of the medicinal plants we are going to proceed. So here there are list of some plants are being given, and a brief information, not like the taxonomic description like we are going to give. Based on the characters with which we can identify the plant, that much of morphological characters we are going to discuss. Then the most important thing is, as I said, active principles are the substances which are responsible for the medicinal property of that plant. Let us say like Raulfia is a very known medicinal plant for all of us, right? So in Raulfia, there are different substances responsible for its medicinal property. As Raulfia is one plant, yields reserpine, that is the substance extracted from the plant of Raulfia roots, that is being used in allopathy medicine also as it is, means without any alteration. Just the isolated substance is being used as a drug. So that reserpine is the major substance, followed by azamelin, azamelicin, and all. So there will be many more lot of other substances also will be there in that plant of Raulfia. But main property of the plant, the drug of Raulfia, is based on the activity of that reserpine. So and also azamelin and azamelicin. So that those are the substances responsible for its medicinal property, and usually found in very smaller concentrations. Those are being called as active principles. Okay, so here we are going to discuss that active principles, and also in brief how they are being used. Medicinal uses of the plants are also we are going to discuss. So in unit 3 and unit 4, we are going to discuss some important medicinal plants. Morphology wise, as a botany student, we need to know about the morphology of the plants, right? That's how it helps us as a botany person to identify the medicinal plant correctly. Identification of the medicinal plant accurately is a very important step in using the plants in medicine. So if there is a lot of ambiguity and confusion, sometimes wrong plants are also being used in either in traditional medicine or even allopathy medicine. So correct identification is the role we can play there as a botany student, right? So that is morphology we are going to learn about certain medicinal plants, their active principles and medicinal uses. That is being given in unit 3 and 4. Then coming to the second block, here we are going to study about the first, that is the fifth unit, we are going to study about the cultivation of medicinal plants. So this is quite interesting and different from the subject we have learned so far. In botany, being the basic science, we try to know about the plants, different groups of plants and how they are being different. And even in the course of time, we try to understand their anatomy, physiology and all that is purely related to the plants. So here we are going to learn about cultivation of medicinal plants. As all of you know, cultivation is an aspect related to agriculture, right? So the agricultural students are going to learn about which crops need to be cultivated in what way and what are the best practices, that is agronomic practices, to improve the yield and the quality of the different crops wise. So that is a professional course where they are going to learn about the cultivation aspects also. But here, being a basic science students of botany here, we are going to include a new thing in our curriculum, that is in our study, that is cultivation of medicinal plants. It is not our sphere actually, but the reason we are including this aspect is that cultivation of medicinal plants is not been followed and also not been cultivated by the farmers in a regular manner like the conventional crops. It is new. Only few plants are under cultivation, and many of the plants are collected from their natural habitats. They are not under cultivation. So there is a need of cultivation for the medicinal plants. So that knowledge being not been known to all the people, so we are also being taking part in that knowing about the cultivation of medicinal plants, because there is a lot of scope is there for the medicinal plants in future. As such, we know there is a lot of market for the medicinal plants right now, and it is going to be increased in future. As we are aware of, the natural products are considered to be more safe. And as the COVID time, we have experienced the use of medicinal plants so much than the allopathy drugs, right? So there will be a lot of scope for the medicinal plants in future. That means the demand will be much more in future. So to meet the demand of the medicinal plants, we need to cultivate them. Unless we cultivate the medicinal plants just by collecting the plants from their natural habitat, we are going to lose that resource forever, or we may just push the plant vegetation to the brink of extinction. Unless you are not cultivating, we cannot use it in such a large scale. So to maintain the, to meet the demands, we need to cultivate. And one more aspect of the medicinal plants is to know about the cultivation is that by growing the plants, we'll know what are the different stages of the plants provides more concentration of active principles, which are more effective. So that means at what time we had to harvest the crop, at what time the active principles are more in concentration, and by also by growing the plants, we can also maintain a quality product all the time required in the drug industry. As all of us know, if it is any of the product is being used in industry means continuous supply need to be there, and also the quality, because being a biological product, it is quite natural. The plants growing in a particular region will have a specific quality. All of us know, like natural product, food items and other crops also, we prefer a particular variety cultivated in a particular region. All of us know that also. Like Sona Masuri, Karnool Sona Masuri we call. That is Varieties sativa cultivated everywhere. Why that specific tag word we are using of the place where it is been cultivated, right? So Darjeeling tea and Simla apple. That means that location has some role playing in that making the product better quality. So here also the same thing will be there. So here it is not being a medicinal plant, it is not our preference to have the best quality or some quality of our preference. Drug need to be of the same concentration of the active principles need to be. So to get that kind of stable amount of the concentration of the active principles, we had to cultivate them. And otherwise, if we just collect the samples from different regions, they tend to vary in the concentrations. The uniformity of the active principles in the drug will not be maintained. So to maintain the quality of the drug, we had to grow them on large scale so that a uniform kind of product will be available, and that will be even supplied to the industry uh, according to the demand it has. Okay, so and also one more aspect of the cultivation of medicinal plants is the most important thing. I always insist to the students also to remember, unless we cultivate the plants, um, we are not going to regenerate them. If we just collect them from the natural resources, we'll not be able to use them for a longer time. So we need to grow them, and more importantly, certain medicinal plants are being collected from the natural habitats. In particular, like their useful parts are underground parts. So if the plant useful part is an underground part, then definitely uh, there won't be any regeneration from the plant. I hope you understood that what is the underground part means. If it is a root, let us say, as I mentioned about the Rauwolfia useful part is the root, and Withania somnifera ashwagandha useful part is the root, and asparagus shatavari the useful part is the root. So when the root is the useful part, we have to pull out the entire plant, is it not? So when you are taken out, pull out the entire plant and remove the root system, then the plant is not able to regenerate again. It's a simple logic. If it is the aerial parts of the plant has been harvested or cut, it regenerates. So here there is no regeneration of the plants happens. So that means there is a loss of the vegetation drastic decrease or loss are going to happen in this kind of medicinal plants. So there are various reasons. So that's why medicinal plants cultivation is an important thing for its conservation and use of the better quality of the plant material and better use of the plants also for the, I mean, sustainable use also is possible through cultivation. That's how the cultivation becomes an important aspect in the medicinal plants study. So here, three plants, medicinal plant cultivation has been suggested in syllabus like aloe vera and andrographis. So that we are going to study. That is about one of the topic. Then the unit 6 deals with the pharmacognosy and adulteration of plant drugs. So here the pharmacognosy is the word is been used in pharmacy also. This is simply about the study of crude drugs. So far we have learned about we are going to learn about what are the different types of medicinal systems, which plants are being used, and how to cultivate the medicinal plants. Certain aspects we are learned. So then comes about the plants use in the medicinal systems. So in traditional medicine, we are going to use the plant product in the crude form. Okay, it's not so that the study of the crude drugs is called pharmacognosy. Even in pharmacy, there is one paper called pharmacognosy. They are going to learn about crude drugs. So the crude drugs means it is nothing but unprocessed drugs. So I just told about the word called active principle, which means the chemical substance which is actually responsible for the medicinal property of the drug of the particular plant. So here it is been part of with other chemical substances of that plant. So it is not never been isolated. The active principles are never been isolated from the plant drugs. So those are the drugs called as crude drugs. Let us say like aloe vera, it will have a lot of active principles. So it is the gel is being used as it is, not been isolating any of the active principle. So that is called a crude drug. So we are going to study about the crude drugs. So which are that crude drugs and how they are being identified and all. And there is also one important study here, adulteration of the plant drugs. So where there is a market, where there is a lot of money, the adulteration is going to happen. That is very human instinct happens in our society, right? So as there is a lot of demand and also the scarcity of certain products also makes the things like that. So there is a chance of wrong substances are being used as a drug, or intentionally some other substance also can be used as a drug. So that is actually been called as adulteration. So that means degrading the quality, debasement, it is also called debasement or the degradation of the quality of the drug is called adulteration. So it can be intentional or unintentional. Okay, so both the things we are going to learn about it. And also there is a method to evaluate. That means to check whether the drug is pure or not. We need to know. Then only we are safe to use the drug, right? So here we are going to use that as an evaluation and adulteration. What are the different types of adulterations happens, and then in what way we can do the evaluation. So whether it is adulterated drug or a pure drug, we are able to do. So that we are going to learn in this pharmacognosy and adulteration of plant drugs. So which is a very important topic in terms of examination point of view also. Then another unit, that is seventh unit, that deals with ethnobotany. So this is a completely new topic for all of us, and also it is having a lot of scope in future for the reason like ethnobotany is something related to the plants use by the aboriginal people or the tribal people or the plants surrounding the native people, how they are being used. So more often we consider it as the knowledge of the tribal people or aboriginal people who are living away from the mainland or main uh, stream society. So they will be having a different kind of the plants use, right? So that is the ethnobotany is the one which is been gaining lot of importance nowadays, because the tribal people are the aboriginal people who are not in the mainstream. They live far away regions, either in the hills or valleys or in the forest regions. They'll be using different medicinal plants for the cure of the disease what they'll get, right? So the plants what they use, and what is the way they use, that means in what form they are using that plants, and what kind of food habits they will have, because the kind of our lifestyle also very much related to the effect of the plants when you are using it as a medicine, right? So mostly in our traditional medicine systems, there will be a lot of food restrictions will be there while we are using some medicines, right? So similarly here, the tribal people or the aboriginal people who are using the plants will be having their own diet and different dietary habits and all. So based on that knowledge, we'll be able to know some more additional new plants, because we always try to find out new plants which we can use as medicinal plants. That search will be always continue, as there is a race happens between the pathogens and the humans. Always the pathogens will be ahead of the humans. Once you reach one point like, yes, we could cure certain disease, they'll be ahead of us, and they try to survive for their like the resistance and all they develop just to survive as a life form. So there will be a race between the drug development and the pathogen ability to survive. That means, for in our context, is a resistance they develop. So our search for the new plants will be always there. So how to search the medicinal plants? Just randomly we can do based on the existing knowledge what we have. Uh, in which families most of the medicinal plants are being included. Based on that, also we can try to explore few more medicinal plants. But actually, the exploring the medicinal plants is not that easy task. We can randomly try, but at the end, it may not give the positive results. It may not give the positive results, even if it is. It appears in our laboratory tests and all, like antimicrobial or anti-inflammatory. What are the medicinal properties it shows? It may not be showing the same positive results in clinical trials, because of the COVID and all of us are aware of all these terms like product development, clinical trials, and how safe and safety measures and all. All of us are aware of that entire process. So here, once even if you are able to identify a new plant with some promising active principle, it may not be showing the same positive result at the end if it is reached even to the clinical trials. But ethnobotany is one area or the field which provides us a ready-made information. That means the people are using that medicinal plants since generations together. That means clinical trials are being done, that is safe, right? So only thing we need to know is what actually that particular plant what they are using consists of. That means phytochemical analysis need to be done. Once the phytochemical analysis is being done, we'll know which active principles will have which type of properties, like whether they will be having cardiac activity or anti-inflammatory activity they will show, right? So diuretic property they have. So all these already we have a lot of knowledge base, no? So based on that we can compare. So only our confirmation of a specific kind of active principles in those plants is the only uh, point will be there. So we can have a lot of that kind of knowledge which will be readily available to us. Okay, so that ethnobotany is going to be a very good source, and also it is recently been acknowledged as traditional knowledge systems. So that is traditional knowledge system. We need to know about it, because most of the time what happens is the people who are been usually in staying in the forest regions or other regions are being relocated, or been they are moving their places in search of like jobs, or even the education wise they are leaving their traditional activities and all. So that traditional knowledge is being lost in different regions, not only in India, in different regions of the world. The traditional knowledge is been not been available to the entire population. I'll explain this way, like all the traditional systems are being documented, they are written. So at any time if you want to refer that, that information will be available. But this information of the plants which are used by the tribal people or aboriginal people is not being documented. So that knowledge is being just passing on from one generation to other generation with the oral knowledge base only. Because unless it has been documented, we'll be losing that knowledge forever. So that is the ethnobotany is one subject is related to sociology, and also it is interdisciplinary. So it is sociology and also anthropology. Then it is also related to different their tribal habits and other things also we need to know. Through that we'll be able to get that knowledge, traditional knowledge, will benefit out of it. At the same time, even the people who are actually holding that knowledge so far also will be getting the benefit of sharing their knowledge through the patents and all. So there will be a lot of scope for the ethnobotany. You can just go through once. It's a quite interesting, and there is even examination point of it. It's a very important topic. Ethnobotany. We are going to know about its history and how it has been came into picture to our study. Then what is its scope and also its importance. Scope means how it can grow further, and importance right now also as an interdisciplinary subject. Then the last topic in the block 2, that is conservation of medicinal plants. So conservation of biodiversity has studied in our first year, taxonomy course 3. So here we are specifying about conservation of medicinal plants. Conservation is the need to make sure of not wasting anything or not damaging anything or protecting. So different words we can use to protect or to conserve, and also to use in a better way. All these things comes under conservation, right? So conservation of medicinal plants we are going to study specifically, because already I told certain points about why cultivation is required of the medicinal plants. So certain things we are going to discuss. You will understand better what is the need of cultivation, what I told by going through the information given here, conservation of medicinal plants. Because the medicinal plants, most of the important medicinal plants are, in fact, they are endemic. I hope you know what is the meaning of endemic. They are distributed in a restricted areas, means their distribution is very limited. So once they are restricted in their distribution, means in a small area when they have been found, and if we are not cultivating them, we are collecting them from their natural habitat. So the loss of that vegetation is going to be very rapid, and they become like endangered, critically endangered, and sometimes even they may become extinct also. So there is need for the conservation of medicinal plants. We are going to learn about that more specifically about the medicinal plants are concerned. Okay, so examination point of view, be thorough with the pharmacognosy topic in this block 2. Adulteration and evaluation both are very important. I repeat, I'll give you the instructions again while we are discussing the huge unit again. Just here I am giving the gist of the topics, and also which are very important and you can go through once. being they are very easy topics to understand. So ethnobotany also study well. Then conservation of medicinal plants. So in the medicinal plants, second unit, block 2, almost all the topics you are able to read little bit and also able to understand. And once I explain, you can also understand better about how important these topics are, not just only examination point of view, as a subject knowledge wise, how important they are. Then I'm moving to the block 3. um, uh, in first year MSc, you have completed about the different plant groups, right? So from algae to angiosperms study has completed. Then anatomy and also the physiology part you have completed. So there is one more important aspect of the basic science of the plants is that their embryology also need to be there. So that is being added to our medicinal plants course 6. So the embryology of angiosperms is another important major part. In fact, if you see the content, it is the major part of having been spread out in 3 blocks. If you see that. So the embryology of angiosperms here, the first unit, that is the unit 9, is deals with the structure of anther and development of male gametophyte. So here you need to recap a little bit of the morphology of the angiospermic plants. Well, you study about the flower structure. We know there will be non-essential organs and essential organs. The flower, I mean to say, the flower consists of like the calyx, corolla, androecium, and gynoecium, right? So the androecium and gynoecium part is being called as essential organs. So the stamens are the androecium. The whole sum of the stamens, everything is called as androecium. So the androecium is representing the male reproductive structures, and the gynoecium is representing the female reproductive structure. So here, as the embryology part, we are going to study structure of anther and development of male gametophyte. So here, you need to have one clarity of what is a gametophyte and what is a sporophyte. I hope you know the difference of gametophyte and sporophyte. There are certain things you need to be very clear about the terms and also their significance. So when we observe the plants, each plant will have a set of chromosomes and all, right? So more often the plants of the higher range, that is the starting from the pteridophyte onwards, the plants are being called as sporophytes. They are in diploid state. Maybe some of the angiosperms are even their polyploids as well, but basic ploidy of the organisms are concerned, they'll have 2 sets of chromosomes. That is called diploid. So the diploid condition in phenotype, it is called as a sporophyte. And their reproductive phase with the is related to the gametophyte. So as all of us know, there are different types of reproduction also we have studied, starting from lower groups to the higher plants, is it not? So that is the vegetative reproduction, asexual reproduction, sexual reproduction, right? The vegetative reproduction is the one which is not involved of any cell divisions prior to the division. That is called a vegetative reproduction. So most of the lower groups of plants like algae, fungi, they reproduce vegetatively, like just fragmentation, buds have been formed. So no special division is been involved at that time of the reproduction. That is called a vegetative reproduction. So even the higher plants are also adapted in certain cases, the vegetative reproduction. If you just think about rose or hibiscus or jasmine, do they develop seeds? Just think about it. They're very advanced plants. When you just think about from algae to angiosperms, the plants which just now I mentioned, they are very advanced plants, but they are not developing any seeds. The reason is they adapted to the vegetative reproduction again. So there are well-developed flowers and andesium and gynoecium, everything is there, but there is no seed formation happens with different reasons, either genetic incompatibility or triploid condition or other reasons as well. And many of the plants, in fact, the ornamental plants, what we grow, they are adapted to the vegetative reproduction. But as we know, the reproduction is one means to propagate their progeny. There is an asexual reproduction and sexual reproduction also. The asexual reproduction is through the formation of spores. And the spores can be haploid or diploid. And there is no fusion of the spores, just directly the spore can develop into a plant. That is being seen in the lower groups of plants, like algae or even in fungi, bryophyta, right? So there is a asexual reproduction there. So they just reproduce through the formation of spores. But there is another method of reproduction. We call it as sexual reproduction, which is a very important reproduction method where it involves the fusion of two haploid gametes and brings the fusion of the gametes and makes a diploid cell called as a zygote, right? So there is one important aspect in the sexual reproduction is that through sexual reproduction, the progeny variation is seen. In asexual reproduction, the progeny is of, because it involves only mitotic division. Mitotic division brings the same kind of progeny. So in asexual reproduction, the purpose of propagation will be achieved, but without any variation. But the sexual reproduction is the one which brings variation in the progeny. So variation is important for the natural selection. With the natural selection only, evolution happens. Without evolution, any species will not survive in nature. There are important phenomena. These are important phenomena in nature. So recombination brings variation. Variation is the basis for selection. Through selection, evolution happens. Evolution is the main thing for any of the species to survive. So there comes the sexual reproduction. Sexual reproduction is so important to bring variation among the progeny. So here we are going to study about the embryology of angiosperms. Here, that is the structure of the anther that is going to produce, as all of us know, anthers are going to produce pollen, is it not? So the pollen is the one which is going to represent the male gametophyte. So here the structure of the anther, mainly anther means not the whole structure. Anther wall is more important because ultimately the anther wall is going to protect the cells which are going to be generated inside. That is pollen grains. Because there is a variation among the different members having different types of anther walls. Okay, there can be 2 layers or 3 layers, and how they are being derived, the layers of the anther also been different. So that we are going to discuss in anther wall formation and then development of male gametophyte. So here the male gametophyte means gametophyte, as already told, gametophyte represents haploid state. So in the higher plants, that is, as we are dealing with the angiosperms, the haploid state in the male gametophyte is represented starting with the pollen grain. Pollen grain forms from pollen mother cell after the reduction division or meiotic division. So the haploid cell formed in the anther in the form of a pollen grain is the beginning for male gametophyte that we are going to study. Then the next one, as being it's a sexual reproduction, is the main thing for the embryo development. So embryology of angiosperms. Also, in the unit 10, we are going to study about structure of ovule and development of female gametophyte. Right. So the structure of the ovule is a very important thing. It is been different among the different plant groups, particularly starting from gymnosperms only. We use the word ovule, not earlier. It is egg up to the pteridophyta we call, right? So here in the angiosperms, there is a defined ovule which is been present in the ovary. There is a difference between the gymnosperms and angiosperms. In the ovary, the ovules will be there. So the structure of the ovule is also an important thing. And from the ovule, how the female gametophyte is going to form. So here the female gametophyte, as I said, the male gametophytes are pollen grains derived from the anther. Here the female gametophyte is the embryo sac. Okay, so the embryo sac is the female gametophyte, which is holding all haploid nuclei inside. Right. So we are going to know about what are the different types of ovules based on their position of the how many integuments are there. These things you have studied in your degree also, but is a kind of revision for you people to know what are the basic structure of an ovule with the integuments and the micropyle and the stalk region chalaza. So whether there what are the different types of ovules like arthrotrophus, anatropus, and campylotropus like that. Then how the female gametophyte is being produced. That is, as I told you, it is a embryo sac. So the embryo sac formation among the angiosperms is also, this is the unique character of the angiosperms. I insist you to realize that embryo sac formation is seen in angiosperms only. Because why I'm mentioning this is gymnosperms are also being called as flowering plants. I hope you know that. There is different classifications like cryptogams and phanerogams. So phanerogams are nothing but flowering plants. So gymnosperms and angiosperms are comes under the category of phanerogams, flowering plants. But there is no flower as such in gymnosperms. And more importantly, there is no embryo sac formation in gymnosperms, except few. I'm not saying 100% it is absent in all the gymnosperms. There are few members of gymnosperms which are having a embryo sac formation for the first time in the plant kingdom. Okay, so that is the examples which you have studied in our first year, like such as netum and ephedra. So the Netials group of gymnosperms, we observe for the first time the development of embryo sac, but it is a general phenomenon in all the angiosperms, formation of embryo sac, right? So that so naturally when the angiosperms are of dicots and monocots, and with so much of diversity among the dicots and monocots as well, we come across different types of embryo sac formation. And basic structure we are able to study, that is the matured embryo sac structure, which is important examination point of view, the structure and what are the different components of the embryo sac we are going to study. Then there are different types of embryo sac formation. That is also important. So that we are going to study as development of female gametophyte. That is nothing but the embryo sac development. Variations of the embryo sac development we are going to study. Then the unit 2 is unit 11 of the block 3 in embryology is related to fertilization. So in the fertilization process, we are going to discuss about the method of fertilization. So what is the fertilization? We are going to discuss. Then there are different aspects. So it is the fertilization we learned from algae to all the, it is been observed in all the plant groups. Nothing new to mention it in angiosperms, but here it happens in a different manner because the complex kind of male gametophyte and female gametophyte had been evolved in the angiosperms. So here in the fertilization process, it is basically the fusion of the male and female gametes or male and female nuclei. That is a fertilization process. Fertilization means, but there is different aspects are being there in angiosperm fertilization. So here we are going to discuss um, pollen stigma interaction. Then there is also, as you know, the stigma is a part of the gynoecium. There is a carpel with the ovary. Then it's a style and stigma. So stigma is the place where the pollen will fall. So in the process of either through wind pollination or through insect pollination, or whether it is a self pollination or cross pollination, whatever, the stigma is the receiving part of the female reproductive structure gynoecium of the pollen. So pollen stigma interaction will be there. So that's where actually the compatibility, if it is of the same plant pollen fallen on the stigma of the plant, then only it will germinate. The other pollen which can also fall on the stigma, but they are not going to germinate there. That is actually the compatibility wise. So there is a stigma pollen interaction will be there, and also the stigma exudates some kind of hormones and also sugars exudates also will be there, which is like chemical signaling, like whether to allow the growth of the germination of the pollen grain or not, or even to allow the growth of the germ tube or not. So all these things will be controlled by the stigma exudate also. Then pollen germination and germ tube growth also we need to discuss, and path of the germ tube, then structure and discharge of pollen tube, and actual gamete fusion, and the types of stigma and style interactions, and finally the most important thing is of double fertilization in angiosperms. So this is double fertilization is the one which makes the another difference between gymnosperms and angiosperms. All of you must have studied this in our intermediate and degree also. But always we need to remember these basic points of the subject. Like, so in angiosperms, there is a phenomenon happens in the fertilization that is called double fertilization. And also another subsequent thing we are going to mention is triple fusion. So here in the embryo sac, there is the egg will be fertilized with one male nuclei, and then the central nuclei also will be fertilized with another male nuclei. That is called double fertilization, and it is also called triple fusion because the central nucleus is central cell will be having two nuclei fertilized with one male nucleus. So that is going to become the endosperm, the development of the embryo. Okay, so that is that makes the difference between angiosperms and gymnosperms. In gymnosperms, the endosperm is haploid, that is derived from the nucellus directly. But here it is the central nucleus with the one male nucleus fusion happens, which is always in triploid state. So three sets of chromosomes will be there in the endosperm of the angiosperms. So this is one remarkable difference between angiosperms and gymnosperms. So the double fertilization we are going to discuss as part of the fertilization topic in the unit 2 of the block 3, that is the embryology part. Then the next unit is the one like the 12th unit, that is called sexual incompatibility. So here the sexual incompatibility is a kind of, as already I mentioned, I hinted that the pollen of any plant, if it is fallen on the stigma of some plant, it is not going to germinate. The stigma is not going to recognize that pollen grain. unless it is of the same species pollen grain. So that is the compatibility. That is, it is able to recognize its pollen, compatible pollen. The other pollen, even if they're fallen on the stigma, they will not be germinating. So that is, in a way, you can also say, you can also call it as incompatibility. They are not able to grow and germinate, and fertilization doesn't happen. So that is actually we call it as an incompatibility barrier. That incompatibility barrier only makes the existence and the identity of the species in nature. If there is no incompatibility between the species, there is no species as such exists in nature. Okay, so this is another thing. You also need to know that what is a species. So many times we use this word species, right? So most of the time the students think if you mention about a genus and species means it is related to some taxonomy. Of course, it is a part of taxonomy. Taxonomy is related to all the fields of science. Whether you are studying about algae or fungi or angiospermic plant, doesn't matter. We are going to use the scientific name, is it not? So that means we are going to use its generic name and the species name. So that species word, also you need to know the definition of that species. What is the meaning of the species? Species means it is a population having similar characters, growing in a particular region, having inbreeding capability. Okay, not having only similar characters is not the word we are going to focus on. Here, a population having similar characters growing in a particular region, having inbreeding capability. They should be able to breed among themselves. That is called a species. So that's how the species is defined. And one species cannot breed with another species. That's what just as just mentioned as incompatibility. That is, incompatibility barrier is a very important thing in nature, is responsible for the existence of so many species in nature. Without that incompatibility, there is no scope for having so much of variation and so many varieties of species of plants and animals also. It's a mix happens, no, all the time. It just a mix, and every time some more some new things are going to happen. It is not happening as all of us are observing. It is not happening. Every species has its own distinctive features. The changes happens in a very slow pace, and a new species may evolve after thousands of years later. So, but there is a time gap to have a stable existence of a species. That is because of the incompatibility barrier. Okay, so that incompatibility is an important thing for existence, and at the same time, there is some negative impacts of that incompatibility if we try to make something new. Like you must have heard, no, the cross between the tomato and potato. If you make natural crossing, doesn't happen. So through different means and ways, we try to make that effort to combine both the characters of tomato and potato. So that is to there are different steps we are trying to overcome incompatibility barriers, just to make sure that our plant breeding experiments and all become success. Okay, so here just the normal method of incompatibility, how it is been operated, the regulatory reform, like pollen stigma interaction, pollen tube style interaction, pollen tube ovule interaction, and the normal incompatibility mechanisms, how they have been operated in the plants like proteandry. That means the androecium matures first, or protogyny, where the gynoecium matures first. If both are at the same time of maturity, then only the fertilization happens, right? So if the pollen matures first. So it is something like that. It cannot. The self pollination is not going to happen. So cross pollination is required. So that means the pollen formed on one plant cannot like pollinate the same plant gynoecium. The other plants, same species. I'm not saying other species. So other plant which is been the early maturity of the gynoecium, there it can pollinate. So that is the pollen protandry, the pollen mature first, then protogyny, the gynoecium matures first, and unisexuality, the flowers itself are making like unisexual flowers. Then diism, like even the male and female plants are different. That kind of incompatibility mechanisms are being operated in nature. So then, advantages and biological significance of self-incompatibility. So what is the advantage and what is its significance? Already I told. By this phenomenon only, the existence of all the species, their stability is being maintained. That genetic uh, composition is being maintained without any alteration. Okay, so there is, as I told you, we try to improve the existing plants of our to improve to add some desired characters in the crops, even to make some intra-specific crosses, intra-generic crosses. We try to make it is our curiosity, and also we try to make the efforts to overcome certain barriers, like incorporating some desired characters in the plants which are not having that character. Okay, so like that, there are methods to overcome self-incompatibility also been tried. That is something like an experimental embryology part, like it will be. So by giving irradiation, but pollen, there are different methods that we'll discuss. So it is basically uh, been the methods been followed in plant breeding methods to generate new type of varieties by combining the two different species characters together, purely for plant breeding purposes. Then that is about the sexual incompatibility of the unit 12 is concerned. Then we are moving to the block 4 of the. Uh, Here we are going to discuss, because in the beginning we discussed about the male gametophyte anther wall, male gametophyte development. Then it is the ovules types and the female gametophyte development. Then the fertilization. Then the incompatible barriers and all we have discussed. Then it is related to the fertilization and all. As I told you, there is one important product is being derived. Not only the zygote is going to develop, as all of us know, the zygote is the resulting cell after the fusion of the male and female nuclei, which is in diploid state. Okay, anyhow, that is going to develop into an embryo. Both in dicots and monocots, it is going to develop as an embryo, right? So, apart from the embryo development, already I told there is a triple fusion that leads to the formation of the endosperm. Okay, so the endosperm is a very important structure in the development of the embryo, the angiosperms, even in the gymnosperms as well. That is the nourishing part for the development of the embryo. Okay, something like a placenta like. So here it is the endosperm which will nourish the developing embryo. So there are different types of endosperm, and also there is even the types of endosperm we are going to discuss how it is being formed, like cellular and like helobial and like that types of endosperm we are going to discuss. Along with the endosperm, there are different additional structures are also being formed along with the endosperm that also we are going to discuss. So endosperm has lot of significance, students, like without the formation of the endosperm, the embryo development will not be completed. Okay, so prior to the development of the embryo, endosperm formation will be initiated, and it will complete the whatever the type of endosperm it will be. It is completed, then the embryo development will progress. So unless the endosperm is formed properly, the embryo is not going to form properly, then the seed will not be viable ultimately for us. So it is endosperm, which is a very important part of the embryo development further after the fertilization. So different types of endosperms we are going to discuss, and additional structures which are related to endosperm also we are going to discuss a little bit. And like the formation we are going to discuss, and also the functions of the endosperm. I just now mentioned in the last, endosperm is the ultimate thing which will decide whether the proper seed, viable seed is going to form or not, because that helps the nourishment of the developing embryo. So the functions of endosperm we are going to discuss. Uh, um, then coming to the unit 14, that is the development of embryo. As I told you, the zygote is going to develop into an embryo. That is the most interesting part. So the embryo, how it is been formed from the zygote. So in the dicots and monocots, the development of the embryo differs. So as we are using the word no, dicot and monocot. I hope you know the difference between the dicots and monocots, not the other characters. The word I mean to say, as all of us know, dicots means it is having taproot system, monocots are having adventitious root system, and also okay, those are all morphological characters wise. The terms what I mean to say, dicotyledons, we know the cotyledons are the part of the seed, right? So there will be two cotyledons that is related to the embryo development and all. So two cotyledons are being there in dicots, and it is only one cotyledon in monocots. So that means we are taking the embryology aspect in dividing the angiosperms into two distinct groups, dicots and monocots. So not just simply on morphology. So we are using, since long time, we are using embryology as one of the aspect in identifying the angiosperms, at least based on whether they are having two cotyledons or one cotyledon. So the embryogenesis also differs in dicots and monocots, and even there are we observe the different types of embryo sacs. As I told you, there are different types of embryo sac development. So same way, even in the embryo development also it differs. So we are going to understand the different types of there are different types of embryo developments in dicots, like onagrade type, asteroid type, like that. So we are going to discuss solenoid type, and so uh, different types of embryo development in both dicots and monocots we are going to discuss. Okay, another important topic after the embryo development and all. So in the unit 15, you can just have a look of it is the topic is apomixis. So far we have just tried to understand in the embryology part the true reproduction wise, how the male gametophyte and female gametophyte have been formed, how the fertilization happened, and what is the net product result of that fertilization in the formation of embryo and the endosperm, how differently they are being formed in different groups of plants of both dicots and monocots, different types of endosperm formation and embryo formation, all these things are we discussed. But there is also another phenomenon been observed in angiosperms, and of course, even the Pteridophyta members also begin. It's apomixis. So it is apomixis is a kind of like without the fusion of the gametes are being formed. It just happened like something like a vegetative reproduction kind. So it is also called as agamospory. That means no gametes are being formed. So apomixis is a kind of feature we observe in angiosperms more importantly. What is the reason for this apomixis? So, genetically, how it is? So, it is the classification of the apomixis we are going to discuss. Like it is only in certain lower groups. Already, I told that in lower groups of plants, they just either like the buds and adventitious branches and all those are things helps in vegetative reproduction. So that is the propagules and all. So embryo formation will be there without meiosis. That means without the fusion, agamospore, as I just told about the apomixis, so it is the embryo is formed without meiosis. So that is called an apomixis. So that is usually been observed in higher plants. So there is something like uh, this is also been referred as the apomixis is also been referred as advent to embryonic. So no fusion of the gametes has been involved, but it develops a kind of embryo-like structure. Okay, so that is been called as advent to embryonic, or sometimes gametophytic apomixis has also been discussed here. Genetically, it has some significance. The apomixis is having some genetic importance, like how it is been formed. Instead of, as I said, no, like without meiosis, it is been being formed, and there is a lot of significance to this apomixis. In fact, without how it is been more than that, how important it is, like the apomixis is being used. Um, for the plant breeding purposes. So, as already told, no, like for the crop improvement, earlier one of the topic I mentioned to you, that is incompatibility barriers. I mean, incompatibility. How to overcome the incompatibility barriers is the one of the major aspect in plant breeding, as I said. And here it is apomixis without involving the fusion of the gametes. So it is almost like a vegetative reproduction, like the somatic characters can be carried to the progeny. So that is been mainly been taken up as a tool for the plant breeding purposes. So here uh, the detection of the apomixis is also one method they follow. But finally, the significance of the apomixis it is a potential genetic factor for use in crop production, crop improvement. So just this is, and even the seedlings which are being formed from this advent embryonic provides uniform seedlings and virus-free clones. So because it's there is no fusion of the gametes and other things, so the most of the hybrids we generate is of a desired somatically desired characters can be achieved. So there is a lot of scope for the application of apomixis in plant breeding that we are going to discuss. One more aspect of the unit 16, that is polyembryony. Must have heard in our degree also about this polyembryony. So we know so far we discussed about the embryo formation after the fertilization, embryo forms, and how different it is also we discussed, right? And without the fusion also there is an embryo formation, that is apomixis also we discussed. But there is another aspect is that sometimes the more than one embryo is been formed in each ovule, that is called polyembryonic. Normally in each ovule only one embryo sac will be formed, and each embryo sac after the fertilization process will turn to be an embryo. That is a regular thing. But more than the development of more than one embryo in one ovule is called polyembryonic. So there is also different types. So as such, there will be the formation of two embryos. That can be one of the reason. Or sometimes that is a true polyembryony has been called. Sometimes the embryonal initials, it forms only one embryo, but the embryonal initials will be split, and that is been called as a cleavage polyembryony. That is one characteristic feature of the conifers. You must have remembered in our course 3, we have studied gymnosperms, right? So gymnosperms, particularly the conifers, exhibits this character that is called cleavage polyembryony. That is one of the things been common for those plants. What is polyembryony? Which is true polyembryony, which is false or pseudo polyembryony, we are going to discuss. And finally, it is about the what is the reasons for it. So the causes of polyembryony also we are going to discuss. But finally, it is the application of polyembryony. So again, it is being used in in crop improvement and in plant breeding. We are going to use this polyembryony aspect. So that is in applied aspect of the embryology wise, you can consider about the polyembryony aspect, how it is being used in the plant crop production improvement and production and all. Mainly application part of the embryology wise you can consider. So the last one, that is actually here it is block 5. We are coming to the block 5, which is related to mostly the applications of the embryology wise. So there is, for every subject will have a different two different aspects, right? While we are doing your masters in botany also has two aspects. One is subject knowledge, and based on this knowledge, what you can do. Okay, application where you can apply this knowledge. So individual subjects wise also, while we are studying or learning, every subject will have some application. That means in our day-to-day life, how that subject knowledge will be useful directly to the man, or in commerce or in economics, how it is been used, how that subject knowledge will be used as a technology, right? So that is called application of the subject. So when we study about the algae also, for a common man it may not be that much appealing, is it not? But if you just give the importance of the algal members as they can be used in the single cell production, and also can be used as algal biofertilizers, and they can also be used for the bioremediation. That is the application part of the subject what we have learned, right? So similarly here, the last block, that is block 5, is going to discuss with the application part of the embryology. So here the first topic is given in the unit 17 of the block 5, that is parthenocarpy. Most of you must be knowing, and immediately you can say that it is without fertilization, it happens in parthenocarpy without fertilization. Or, and more often, we also relate this phenomenon with the development of seedless fruits. Is it not? So the parthenocarpy is the one which is been observed in nature, and that is being taken up by the plant breeders as a tool or a technique to develop seedless fruits. We are getting many kind of seedless fruits, either it is the grapes or even the pomegranate. The pomegranate seeds of the native varieties or the wild varieties earlier, it used to have very hard seed coat. The pulp is too less and the seed coat is too very thick. But now we are getting more of pulp, and almost the seeds are flat with thin seed coat. So they are not even able to germinate. They are not viable seeds also, right? So seedless varieties of the different kinds of fruits we are able to develop through the study of parthenocarpy. It is induced parthenocarpy we are using as an application wise, which has a lot of significance in the development of different kinds of fruits, mainly. So here in the parthenocarpy, we are also dealing with, as I already told, it is the application part of the embryology. So here certain topics are being given, like here, yeah, how it has been used. And then the other aspects of the this fifth block is also included the application of different aspects of embryology, such as it is directly been given as experimental embryology. So far we have discussed like how the apomixis can be used, how the polyembryony can be used. So direct techniques wise, how we are able to use the embryology, that is experimental embryology we are studying in the unit 18. Then coming to the unit 19, it is applications of embryology in taxonomy, agriculture and horticulture. Okay, applications of embryology and taxonomy that we have studied in our first year of the course 3 also, because there are certain instances of different types of embryo sacs. If you just once we go through the different types of embryo sac, we are going to identify each type with one genera, or sometimes with one, mostly with the one genera we are going to identify. That means it is having a lot of significance in systematics. That particular type of embryo sac is found only in that genera or only in that family. So that distinctiveness makes it is a significant tool in the systematics for the identification of different genera and different families. So, and sometimes even the embryological data also helps in solving some problems of distinguishing two very much identical genera or very much identical species. So those are the examples we are going to study in what way this subject of embryology is helping in taxonomy. Then agriculture. Already we have told about the applications of embryology in agriculture. As I told you, in plant breeding, we are going to use a lot of um, aspects of the embryology. So that is in agriculture and horticulture. Just now I told seedless fruits we are getting, and also the kind of seeds fruit size need to be improved, and what are different characters wise we are going to introduce that in horticulture, and even in the cultivation of the plants wise also. So if the plants are not been developing the viable seeds and all, so in horticulture we are able to use our embryology knowledge to develop seeded varieties, or if they are not, so some recently you must have observed hibiscus normally is a vegetatively propagated plant. If you just happen to know about some of the hybrid varieties of hibiscus, they do develop seeds. So by developing the seeds, we can also make the crosses between the different types of hibiscus varieties, and can improve different types of flowers, not by vegetative grafting, by having the seed development introduced there, and through which we can develop true hybrids kind of variations we can bring at them. So that is also been played an important role in horticulture to develop different kinds of ornamentally valued plants. Okay, then the last aspect is principles of application of palynology. So in fact, this is been included in the last, but palynology is a subject been related to the study of pollen grains. Okay, so that is a very important topic, in fact. So that palynology study, applied aspects of the palynology wise. So how it is being used, the palynology study, how it is being used. Like it is also called as like recently we are many of us are using more of honey in recent times. So we know there is a single varieties of either it is neem honey or mango honey, tamarind honey, different honeys we are getting the brands wise, not the companies names I am talking about. So that means we can identify the honey been derived from which kind of flora, based on the study of the pollen grains. The honey consists of pollen grains. So the based on the pollen study, we can identify whether it is having only one type of vegetation being producing that honey, or different plants are being, even whether it is uh, to assess whether it is pure or not. Also, we can use the palynology, because if it is not having any pollen grains, we can show that it is not at all a pure honey. It is a sugar syrup kind of thing, colored sugar syrup. So even based on the pollen types, we can say like whether they are that pollen is an indication of that plant's existence in the surrounding areas. So based on that, we can also assess whether it is been collected, that honey is being made in our area or been produced in a different faraway regions, whether it is pure or impure, having many pollen grains. So that is basically it is called as melitopalenology. So the honey analysis based on pollen is a very important tool nowadays. They are using to make the standards of the honey. And pollen in medicine is also been used because pollen is considered to be highly proteinaceous. So in Western countries, they use pollen tablets also as a protein substitute. Here it's not that much important, but it is being used. So pollen in medicine is also been used, and also pollen in taxonomy been used, and pollen is also being used in forensic science. So there are different aspects of the application of the paleontology. As a part of the embryology wise, we are going to discuss that in our course 6. If you follow this recording also, whenever it is convenient to you to follow the recording, you can follow. And if you join the class according to your convenience, that is the best option. So that we can also have some kind of interaction as well. You can raise your doubts. I'm concluding here about just I try to discuss about the syllabus, what we have in our course 6 of having medicinal plants and embryology of angiosperms. Tomorrow's session, I'll start with the medicinal plants discussion. Being it's a Sunday, try to join the session so that we can have a direct learning class, and also you can interact. You can interact and you can raise any doubts if you have, or related to the course or related to the topic discussed during the time session of the time. Thank you.